And Christy, do you care to come on up here? Okay, really glad to have you here with us. Christy came all the way here from St. Louis, Missouri, and it's a long drive. Glad to have you here. Appreciate that on the lights. Christy E. Edwards, PhD, received a BA degree in ecology and environmental biology and Spanish from the University of Colorado at Boulder, and a PhD in botany from the University of Florida in 2007. She was a postdoctoral researcher in the Department of Botany at the University of Wyoming, and then worked for 2 years as a research biologist in environmental laboratory of U.S. Army Engineer Research and Development Center. Dr. Edwards joined the Missouri Botanical Garden in 2013. She is the Department Center. Dr. Edwards, let's see, hang on just a minute. Okay, let's see, with Missouri Botanical Garden in 2013, and she is currently at the Stephen and, if I'm saying this right, Camilla Breuer Conservation Geneticist, and the Missouri Botanical Gardens and Laboratory, and Adjunct Professor of Washington University in St. Louis, Missouri, and the University of Missouri St. Louis. Her primary role is the lead for the conservation geneticist research, and a lot of good stuff. Programs at the University and the Botanical Garden. Her research focuses on using population genetics, quantitative genetics, molecular systematics, thank you. Need more coffee. And DNA. Very good. Yeah. Okay, to help understand the ecology and evolutionary biology of endangered species, and it's applied for in situ and ex situ conservation efforts. Okay, and so we'll go ahead and leave it to you. Thank you for being here with us.
Thank you. So,
she's very well qualified. Okay, I think. Kim, we need to give her a microphone.
Where is it? Oh, it's right here. Okay, you can hear me. Yes. Okay, there's one on the podium. Okay, so thanks, Steve, for the invitation, and thanks for everybody for coming to listen to me talk. So today what I'm going to be doing is talking about research that we're doing at the Missouri Botanical Garden into genetic and phenotypic diversity in the Ozark Chinquapin. All right, so to start off with, I'm going to give an overview of the larger study that I'm working on. So the reason I'm working on this Ozark Chinquapin is actually because of this larger study that was funded through a grant from the Institute of Museum of Library Services, which is a federal agency. The Botanical Garden is actually considered to be a museum. And the idea is we're trying to address a problem that people in the Botanical Garden community face. And essentially that problem is trying to make sure that we're conserving diversity of plants in our collections. So just keep that in mind. And this is a relatively new study, so I just wanted to let you know that right now basically I'm talking about what we're doing. I don't have a lot of results yet, but hopefully I can come back in a year or two and tell you about the actual results of the study. Okay, and then, so first I'll talk about the overview of the main study, and then I'll just talk about the progress that we've made in Ozark Chinquapin. Okay, so as you are well aware, and as the two previous speakers have talked about, biodiversity is declining. So the current plant species extinction rate is about 500 times the background rate that happened kind of before human settlement. And about 42.8% of plants are considered to be threatened or endangered with extinction. And the cause is generally competition with a single species, which is us. We do things like destroy natural ecosystems, use them unsustainably. We import invasive species like blights and also other species that are competing with plants, like, for example, the bush honeysuckle. And we do things, and we're also, there's climate changes happening too, so that's something that we're going to have to be thinking about going forward. So because of that, we, as we all in this room know, conservation is really important. And I would say that, like, in general, probably the most important way to conserve a species is through in situ conservation, or conservation of a species in its natural habitat. And this includes both conservation of the land, but also land management activities like burning and prescribed fire, removal of invasives. But there's a lot of species that you can't necessarily conserve in the natural habitat. Or maybe you want to have a backup, in which case that's what in situ or ex situ conservation is essentially conservation outside of a natural habitat. And that's what we do with the Missouri Botanical Garden. That's what the plots that Steve has, where he has the Chinquapin plots. These are all ex situ sites. In general, in botanical gardens, the majority of ex situ collections are bank seed that we take and we dry it down and we freeze it and we store it in a freezer for our ex situ collection. But there's a lot of plants out there that don't actually freeze well, and they won't. The seeds won't revive after they've been frozen. So in that case, you have to conserve whole living plants. And a great example of that are Ozark Chinquapins and oaks and other things like that have these nuts. They don't stay viable in a freezer. And so because of that, you have to have living plants in a botanical garden or in a plot. The idea of these is to safeguard a species against extinction. And the idea is you're sheltering a representative sample of diversity from threats that they may experience in their native range. And then it's also used as material for reintroductions, population augmentations, just like what Steve is doing, where he's taking material and sending it out to people to plant into their natural areas. I would say, though, that because it serves as a backup and it's so important, one of the things we want to do is make sure that we're trying to conserve as much diversity as possible in these collections. And ideally, all of the diversity. So that's a big question, right? Do these collections actually effectively and efficiently represent the diversity present in a species? So effectiveness is the extent to which diversity is conserved. And I think, if you have an ideal situation, maybe you could collect seed from every single individual in the population. Obviously, that's or in a species. Obviously, that's pretty much not feasible for most species that are endangered. And so the question is that when you have less than ideal conditions, how do you make sure that you're conserving all the diversity? We're also talking about the efficiency. So, things like Ozark Chinquapins, like in a botanical garden, we can't conserve 1,000 plants, right? So we want to make sure we're trying to conserve as much diversity as we can in the smallest number of individuals possible. And that's just because space is limited. We can't conserve 1,000 trees. So the question is, can we maintain as much diversity as possible by maintaining a much smaller number of individuals, like maybe 100 or something? So there's different ways to do this. One of the ways that is standardly used in the botanical gardens or in other places is to use the CPC best plant conservation practices. And the idea is there's a set of guidelines, and essentially there's actually a whole book about this. I'm not going to bore you with all the details, but you do things like you want to collect from 50 plants and up to 3,000 seeds. You collect from 5 or more populations from throughout the range. The list goes on and on of things that you're supposed to do. But there's been several studies where they've assessed the efficiency of these collection protocols. And what they found is in some cases it works, and sometimes it doesn't, largely because of the way the organisms pollinated, the way the seeds are dispersed, and the way the genetic diversity is structured across the landscape. So there's other ways to do this. So I'm a conservation geneticist, and this is a lot of what I do. This is a situation, this is the Physaria filiformis, or the Missouri bladderpod. And for this, what we did is we went out to every population that we could find. We sampled individuals, we brought them back to the lab, and we genotyped them. And then we looked to see how genetic diversity at neutral loci, so loci that are genetic markers that are not under selection, how the genetic diversity is structured across the landscape. So here what we see is these colored circles right here. So each one of those represents a population, and the color represents a genetic group. So to make a recommendation for conservation for this species, what we did is we said we want to protect, conserve in situ and ex situ one population from each one of those population groups, so one of each of the colors. However, this is only at neutral genetic diversity, but there's actually other types of variation out there. So there's not only neutral genetic markers, so these are ones that aren't under selection, but there's also ones that are under selection. So for example, like, in North America, we have a climate gradient, right? It's colder up north and it's warmer down south. And so if a species has a relatively wide range, they can experience different conditions across that range. So for example, in northern populations, they may experience very cold, and so they may be cold adapted. In southern populations, they may be heat adapted. And because of that, you can see a signature of local adaptation across the range of the species. And the way that you can test this is actually to take genetic markers, so like you'll have all the individuals in the genotype, and then you can look at the allele frequencies of that genetic marker, and you can associate it with environmental variation. So, for example, here on the right, you can see where it says neutral gene. There's an association here, and we see that there's no association between gene frequencies and environmental variation. So that's a gene that's not under selection, it's behaving neutrally. In other genes, what you can see is there's a strong association. This graph here on the right, I don't think this, you can't really see that, on the right, and essentially you see a strong association between environmental variation and gene frequencies. So this is a gene that may be showing signatures of local adaptation. So that's how you can kind of differentiate between genes that are under selection and genes that are not. The same can be said for phenotypic traits. So we have phenotypic traits that you can go out and measure, like plant height. Light resistance is a great phenotypic trait. And so what we can do is we can look at the values of individuals across a environmental gradient and see if that trait is associated with environmental variation. And on the left here, you can see there's no association. On the right, you can see that there is an association, so a strong association between the phenotype and environmental variation. So this is kind of a way to get at what we're getting to get at the variables that we're measuring in our study. So the reason why I'm saying this is because the different forms of variation can become uncoupled across the landscape. So this is a study where they took genetic loci and they divided them into neutral markers and adaptive markers, and then they looked at how they were structured across the landscape. So on the top graph, you see they have 3 genetic clusters, the red, yellow, and blue, and that's neutral variation. In the bottom graph, they looked at the same individuals, but with different markers, and they found 6 groups. And you can see it's a very different pattern. And so, for example, if you want to conserve the variation, in the top you may only want to protect 3 different populations. On the bottom you want to protect 5. So the strategy differs. And this is important because we all have limited conservation resources. So say you only want to protect 2 populations, then the strategy can be very different for how you want to conserve that variation. So this is a study that was conducted in 2007 where they went out and they sampled a bunch of populations, they genotyped them, and they measured their adaptive variation and their neutral variation. And then they plotted them on this graph. So in this situation, they said we can only afford to conserve 2 populations. And so if you wanted to conserve the the two most adaptive populations, one that have the most adaptive variation. You might protect the RM, and if you can see that these ones right here with the dotted line. But if you wanted to protect the most neutral variation, you might want to protect these two populations right here. And if you wanted to protect the populations that had the most neutral and adaptive variation, you would want to protect the RM and PP populations. So the strategy differs. This is another way of looking at this. So this would be taking individuals and genotyping them and then measuring like their adaptive and neutral variation. So for example, so on the x-axis we have the amount, or the amount of genetic diversity right here. On the y-axis we have the amount of a desirable property, say blight resistance. And there's some species where you may be able to conserve a lot of the desirable properties, say the blight resistance. So this curved line at the top, while still protecting diversity, like neutral genetic diversity. And there's other species where you have this line where you may be able to conserve a lot of, for example, blight resistance, but you might not be able to conserve a lot of the neutral genetic diversity, and vice versa, assuming that you have a limited collection size. And essentially that's what we're going to be doing in this study, is looking at whether there's a trade-off that exists between conserving neutral versus adaptive variation. Okay, so generally the overall study questions that we're working with here are, do we see local adaptation in imperiled plants? That's just a kind of a basic question, and people haven't really investigated it. The second is, if so, are neutral and adaptive variation in phenotypes and genomes, are they structured differently across the landscape? If so, assuming a limited collection size, do trade-offs exist in our ability to protect all of the forms of variation that exist in these populations? So the main goal is to try to get all of the diversity into a small population, and so that's kind of the idea, is we're trying to understand the dynamics under this scenario. Okay, so overall, like I said, this is a larger study. So we're focusing on 3 herbaceous species, including Boltonia decurrens, Physaria globosa, and Dahlia foliosa. All 3 of these species are federally listed. They do occur, they have a pretty long latitudinal gradient of their population occurrences. So it's kind of increasing the likelihood that they are going to show local adaptation in response to environmental variation. And then we have 3 woody species. One is the Ozark Chinquapin, another one is Quercus arkansana, and the last is Navusia alabamensis. So Navusia doesn't produce seeds in its native range, and then the 2, Castanea and Quercus, don't produce seeds that can be banked. So all 3 of these species represent species that have to be conserved as whole living plants to be conserved in an ex situ collection. So that's one of the reasons why we're doing this, because we actually have collections of these species and we don't want to conserve like 600 of them. So the overall experimental design is that we're trying to leverage resources that are available in many botanical gardens, because this is supposed to be applicable to other botanical gardens or other people doing ex situ conservation. So the idea is we're using our living collections as our study material. We have our growth facilities. So we have a large greenhouse that we're using as a common garden to do measurements of phenotypic traits. And then in my conservation genetics lab, we're doing all the genotyping of all the individuals. So basically what we've done is we're growing all these individuals in our growth facilities. Then we're measuring them both in terms of phenotypic data and genetic data. And then we're going to conduct associations with the environment to differentiate between neutral and adaptive traits, like I said before. And then we're going to assess trade-offs in the conservation of different forms of intraspecific variation using an approach called simulated annealing. The idea is that then we're supposed to be like leading the field of botanical gardens so that they can do a good job of conserving diversity in their collections as well. Okay, So let me just talk briefly about some progress in Castanea ozarkensis. So for those of you who have worked with Castanea for a long time, one of the things that you know is that there's some issues with taxonomy in terms of Castanea ozarkensis and Castanea pumila or Allegheny Chinquapin. So this is the maps, these are range maps from BONAP, and as you can see they overlap quite a bit. And there's actually been some genetic studies done. This is a study that was published in 2020. They didn't sample a lot of Ozark Chinquapin, but they did sample most of the Castanea pumila and ozarkensis across its geographic range. And what they found is that there are some populations that appear to be pure Castanea ozarkensis. Those are the ones in red. And then Castanea pumila is supposed to be in like the purple, blue, and light blue. But as you can see on the graph at the top, so the individuals that have a full bar of red are ones that are supposed to be pure Castanea ozarkensis. The ones that are light purple are fully light purple are pure Castanea pumila. And then there's some individuals that are intermediate, where they're half light purple and half red, and those are hybrids between the two. However, so there is hybridization between the two species.
What's the brown one?
The brown one?
Yeah.
Oh, that's also Castanea pumila.
Okay.
Yeah. So I'm just talking about the ones that are hybridized. Yeah, so all the rest of them, so the light brown, the light blue, and the purple are all supposed to be Allegheny Chinquapin. The red is Ozark Chinquapin. And then you can see there's hybridization between the light purple and red, which are essentially these bars right here, which you can't really see this. Anyhow, so they didn't sample a lot of Ozark Chinquapin in this study. I mean, I think they had like 6 or 7 accessions. So one of the things that we want to do in our study is to do a much better job of trying to figure out where the boundaries are between these two species in the southeastern U.S. So that's kind of one of our first questions, is are they genetically and phenotypically distinct? Do they hybridize? And if they do, basically we can take those individuals and kind of put them aside and then only focus on the ones that are Ozark Chinquapin. Then, once we have those individuals, then we can look to see if we see evidence of local adaptation, and then whether we find trade-offs in the conservation of adaptive and neutral variation, both in phenotypes and genotypes. So, what we've done so far, actually what Steve has done mostly, is we essentially approached him and said, this is a study organism that we'd like to work on. We didn't have an ex-situ collection when we started this project, so we talked to Steve and we said, we'd love to do a big genetic study on this, we'd do all these phenotypic measurements. And he kind of mobilized this whole group of volunteers who came and collected all these seeds for us, and Steve delivered us like 1,000 seeds or something. We ended up with over 600 viable seeds from all these different areas, or kind of across the range of Castanea ozarkensis, and then there's some Allegheny Chinquapin in there as well. So then we have all these seeds, and they're now growing in our greenhouse. So what we've done so far is we've essentially germinated them, and we're measuring them on a monthly basis for things like germination, emergence, stem height, number of leaves, leaf length and width, and stem diameter. And these things are just, like you can see the pictures, they're about this big at this moment. And so the idea is that, so we have these phenotypic traits, and we can do some preliminary analysis to see if there's some evidence of local adaptation. So we're doing associations with latitude at this stage. So what we've seen so far, so this is just a graph showing an association between a couple of phenotypic traits and latitude. In the red, we have Castanea ozarkensis. In the blue, we have Castanea pumila. And so what we can see is that we see some local adaptation, and it differs between the two species. So in red on the left is the stem diameter. We can see there's a positive association where the plants at more northern latitudes have larger stems, and in the Ozark Chinquapin, and the opposite is true in the Allegheny Chinquapin. And then in stem length, we also see that both species are negatively associated, so the shorter plants are from the northern part of the species range. But the slope of that line differs, so there's a much stronger association with climate in the Allegheny Chinquapin than in the Ozark Chinquapin. We also see that some traits don't vary according to latitude. So for example, this is the leaf length, and we see that neither one of the species are associated with, the trait variation is not associated with latitude, and leaf width is the same. So we see that some traits are showing evidence of local adaptation, others are not. So that's actually good, because that's kind of what we're trying to find out. Right now we're also in the process of genotyping the Ozark Chinquapin. So we've taken these samples from all of the 600 individuals that we have in our living collection. And the one thing we have done so far that we've completed is we've looked at genome size. We've taken 2 samples, one from Ozark Chinquapin and one from Allegheny Chinquapin, and looked at the genome size. This is just so that we can design our DNA sequencing experiment. We're in the process of building a high-quality reference genome for the Ozark Chinquapin. And then once we have that, then we're going to be doing genotyping using low-coverage whole genome sequencing. And then we're going to take that data, analyze genetic structure, and try to analyze the species boundaries to try to differentiate between Ozark Chinquapin, Allegheny Chinquapin, and hybrids. And then once we have the data from the Ozark Chinquapin, then we'll conduct associations with latitude or climate to test for local adaptation, and then identify neutral and adaptive genes. So the one thing that we have found is that we have samples from Castanea ozarkensis and Castanea pumila, and we actually see quite a large genome size difference between the 2 species, so that's another thing that differentiates them. So Castanea ozarkensis has a genome size of about 1 picogram, and Castanea pumila has a genome size of 1.4, which is significantly larger. So there are more differences between the 2. So in general, what we found so far is that the 2 species differ in genome size, and they also show contrasting patterns of local adaptation in some phenotypic traits. Stem traits show local adaptation in the Ozark Chinquapin, whereas leaf traits don't seem to. And then northern Castanea ozarkensis have wider, shorter stems than southern populations. And then, like I said, this is just the beginning of the study, so we're actually in the year 1 of a 4-year study. So this is just the really preliminary results. Hopefully I'll come back and tell you more about the results. But generally, the first thing we're planning to do is to generate the data to clarify the species boundaries, and then look at local adaptation and trade-offs in our collection. We'll reanalyze phenotypic traits once we know which individuals are Allegheny Chinquapin and which ones are Ozark Chinquapin. And we'd like to phenotype additional traits. Probably the most important one will be light resistance, but also things like physiological traits and reproductive traits once the plants start flowering and fruiting. Then we'll analyze trade-offs in conserving different forms of variation, and try to make conservation recommendations based on those results. So overall, I think the future implications is that, first of all, we'll try to resolve the limits and status of Castanea ozarkensis relative to Castanea pumila. We'll clarify how intraspecific variation is structured spatially, so that we can make sure that we do a good job of conserving it. And then that will have important insights into trying to make sure that we conserve that variation that exists out there in the natural populations. Okay, so I'd like to thank Steve, the Ozark Chinquapin Foundation, and all of the people. I don't know if any of you guys have sent me seed, but if you have, thank you for helping us build this collection so that we can do this study on Ozark Chinquapin. We're funded through the Institute of Museum and Library Services, and then I have a whole bunch of people who have helped out with this project. So with that, thanks.
One more question. So I've grown a lot of chestnut seeds from New York, and I've also grown a lot of chestnut seeds from Virginia.
Yeah.
And you see, well,
Yeah.
And I was wondering if you have seen the same thing. It makes sense to me because you're in a colder environment.
Yeah.
You don't want your seed to germinate at a time where you're going to frost.
Yeah, we saw huge variation in seed germination time. Like, I mean, it was like, there's some that were germinated when we got them, and then there's other ones that were germinating like 4 months later. So actually, like, one of the things that we were struggling with is that, like, you know, we were trying to do phenotyping, and half the population, like half the seeds hadn't even germinated yet. So this year, in some ways, we were a little bit like, well, I don't know about all the phenotyping data we have because all the individuals kind of started at different times, you know. But next year, everybody will have been germinated and have a single like growing season behind them. So hopefully they'll be a little bit more even in terms of their developmental stage.
Yes, sir. Go ahead.
As a practical matter,
Turn it on. Get the bottom.
It's the very bottom. There you go. Push it in. There you go.
Hello?
you turned it on.
Things that make me say, Okay, go ahead, speak up.
Just yell.
I know it's in a very preliminary stage, but have you been able to discern any difference in blight resistance between cumulent and ozarkensis?
So all of our plants are in, so we haven't measured blight resistance yet, just because the plants are so young. And also, I mean, yeah, so we haven't done it yet, but that is definitely something that we want to measure. Like I said, this year we had a huge variation in germination time, so, you know, a lot of these plants have like 3 or 4 leaves on them, and so we didn't want to like start chopping up the leaves yet. We kind of want to wait until they're a little bit like more robust. So we don't know yet, but we will find out. So stay tuned. Hopefully I can come back in a year or 2 and tell you about this. And actually, I didn't know about the assay where you could just soak things in, what's that? Like the...
Oxalic acid.
Yeah.
Oxalic acid.
Oxalic acid. We didn't know, so that's much
It's
easier.
really pushed everything ahead for us.
Yeah, because, so we were thinking, Leslie initially did these like blight resistance screening on leaves using the actual blight. And we don't really want to do that because we don't want to infect our population yet. So anyhow, that's a great...
And you know, we've come a long way. What she's talking about is originally what we did what's called leaf blight assay, where we actually took the blight, Cryphonectria parasitica, and injected it into a leaf, and to see how it would react. And it takes a little bit longer. And then, like you said, you've got to have the blights. We were handling blight doing this, you know.
Yeah.
And so it's kind of like, you know, playing with dynamite. You know, well, let's see what happens here in a controlled situation. What could go wrong, you know? So anyhow, so we've come a long way. So the oxalic acid test really shot us ahead, I mean, big time. And so Christy had mentioned a little bit about this too. So what we did, we sent her seeds on the map you saw from what we had. We actually now have, and I've got them in 5-gallon buckets, and we got some cat litter that you can quick dry these leaves immediately, and we hadn't even got to that yet. And so this is just the beginning initial results she's having from this study, and it takes, like she said, 3 or 4 years. So hopefully she'll come back again. But these leaves that we have leaves from all the way out to Virginia of Chinquapin trees. I'm just going to say Chinquapin trees. I'm not going to say if it's Alabamasis or what it may be, but it's Chinquapin trees, and then you have a Chinquapin bush. That's the old way they used to identify these two. And so anyhow, with these leaves, we quick dried them as quick as we could get them in. And so from those, she can extract, the quicker we did this, there's more, and correct me if I'm wrong, more genetic material you can extract from them. So going back to what you were asking, there's probably markers in there about, you know, different things that she can find. And from another molecular study that was done, the markers were off. And we had one of our great big Chinquapins, and they identified it in the molecular test as actually being a Chinquapin bush. So we're taking that information, what she's got. This is going to be groundbreaking, and it's going to really, really help us fine tune down. So if we have a population, maybe in North Carolina, you know, she will give us the markers for that population, but it's not going to happen yet. She hadn't even got from me yet
Yeah, we
these
need to get those
leaves.
leaves.
Yeah,
Actually, we
so
need
we've
those.
got plenty of those, so I got to get them up to you.
Yeah.
And so anyhow, then we're also raise your hand if anybody here actually turned in a leaf sample or leaves, raise your hand up.
Yeah.
Okay,
Thank
we've
you.
got people in here that did it. And so what we're actually going to have, and this is kind of a neat thing, so you can go to the Missouri Botanical Garden website and click on a link, and then I've got a big stack of herbarium specimens that are dried, and they're going to go ahead and hang those for us, and it will have that finder's name on there where it's at by county. We didn't give specifics, and it will show the range of the trees where it's actually at. And so there's a lot of pages waiting to be filled, and we're just proud that you're able to do that. That's really good, this partnership.
Yes.
They actually hired more people to do this project for these 6 species, and the Ozark Chinquapin is one of them.
Yeah.
And so this is, folks, this is going to take 3 or 4 years for all the results to come in, and this is just preliminary to what she's found so far, though. So we just really thank you, and I think this is awesome.
Yeah.
And I think we need to give her another hand. And time to take a break.